Welcome to G Fun Facts Online. Today, we're venturing into a world that is closer than you think. In fact, it's inside you right now. Imagine a bustling, invisible metropolis teeming with trillions of residents, all living and working deep within your digestive system. This isn't science fiction. It's the human gut microbiome. For a long time, we thought of these bacteria as simple helpers for digestion. But we're now discovering they are so much more. They are silent partners, powerful chemical factories that have a profound say in how our bodies process medicine. This realization has launched a revolutionary new field of science called pharmacomicrobiomics, and it's completely changing our approach to health, promising a future where medicine is tailored not just to you but to the trillions of microbes you carry. So let's dive into how these tiny partners are shaping the future of medicine. For decades, the undisputed champion of drug metabolism in the body was the liver. We pictured it as the central processing plant, the organ responsible for breaking down, activating, or deactivating the medications we take. And while the liver is absolutely a metabolic powerhouse, we now know it has a partner, a second highly influential organ we never really accounted for, our gut microbiome. Think about the scale of this. You have about 20,000 human genes in your genome, but the collective genome of your gut microbes, the microbiome, contains millions of genes. This gives them an immense and diverse arsenal of enzymes, molecular tools that can perform a staggering range of chemical transformations on the drugs passing through our gut. In essence, your gut bacteria function as your body's second liver, but one that is incredibly variable from person to person. My microbial liver is different from yours, which helps explain why the same drug at the same dose can have wildly different effects on different people. But this relationship isn't a one-way street. It's a complex feedback loop. The drugs we take can, in turn, dramatically alter the landscape of our gut microbiome. Common medications you might not even think about, like proton pump inhibitors for acid reflux, the diabetes drug metformin, and even some antidepressants, have been shown to shift the balance of our gut bacteria. This creates a dynamic interplay where the drug changes the microbes, and the changed microbes then alter how the drug, and maybe even other drugs, will work in the future. So how exactly do these microscopic residents meddle with our medications? They have a sophisticated toolkit of strategies. Let's break down the four main ways your gut bacteria can control the fate of a drug you swallow. First up is drug activation. Some medicines are designed as prodrugs. This means they are administered in an inactive form, like a secret agent in disguise. They need to be activated in the body to become therapeutically effective. Often, we assume the liver does this job, but in many cases, it's our gut bacteria that perform the crucial activation step. A classic example is a medication called sulfasalazine, used to treat inflammatory bowel disease, or IBD. When a patient with Crohn's disease or ulcerative colitis takes this pill, it travels all the way through the stomach and small intestine largely untouched. It's waiting for the right signal. When it reaches the colon, where the bulk of our gut bacteria live, specific microbes produce enzymes that act like molecular scissors. They cleave a specific chemical bond in the drug, releasing the active anti-inflammatory compound right at the site of inflammation. It's an incredibly elegant system, using our own bacteria for targeted drug delivery. The second mechanism is the complete opposite: drug inactivation. Just as they can turn drugs on, gut bacteria can also switch them off, sometimes rendering them useless. The most famous case of this involves digoxin, a medication derived from the foxglove plant that has been used for centuries to treat heart failure. Digoxin is powerful, but has a very narrow therapeutic window. Too little is ineffective, and too much is toxic. For years, doctors were puzzled why some patients required much higher doses than others. The answer was found in a specific gut bacterium called Egertella lenta. In about 10% of people, this microbe is present in the gut, and it essentially eats digoxin for breakfast. It metabolizes and inactivates the drug before the body can absorb it, leading to dangerously low levels in the bloodstream and a lack of therapeutic effect. Identifying this bacterial culprit was a landmark moment for pharmacomicrobiomics, showing that a single type of bacteria could be the difference between a life-saving treatment and an ineffective one. Now for the third, and perhaps most concerning mechanism: toxicity modulation. Sometimes, the metabolic activities of our gut microbes can transform drugs into toxic byproducts, causing severe side effects. The textbook example here is the chemotherapy drug irinotecan, used to treat colon and other cancers. The body has a clever way of handling this potent drug. The liver takes active irinotecan and attaches a molecule to it, a process called glucuronidation, which essentially puts the drug in safe mode, deactivating it so it can be safely excreted. This inactive, safe form of the drug then travels to the gut. But here's where things can go wrong. Certain gut bacteria produce an enzyme called beta-glucuronidase. This enzyme does the exact opposite of the liver. It snips off that safety molecule, converting the drug back into its toxic, active form right there in the intestines. This leads to severe, dose-limiting diarrhea and intestinal damage, which can sometimes force doctors to stop a potentially life-saving cancer treatment. It's like the liver is a bomb disposal squad carefully disarming a bomb, and the gut bacteria are coming right behind them and rearming it. Finally, there's a more subtle mechanism called drug sequestration or bioaccumulation. Here, the bacteria don't necessarily change the drug's chemical structure. Instead, they act like tiny sponges. They soak up the drug, storing it inside their cells. This process effectively traps the medication in the gut, preventing it from being absorbed into the bloodstream where it's needed. This lowers the available dose, potentially reducing the drug's overall effectiveness without anyone realizing why. These direct interactions are fascinating enough, but the influence doesn't stop there. Our gut microbes can also have powerful indirect effects. They produce thousands of different metabolites that can enter our circulation and travel throughout the body. These microbial signals can communicate with our liver, telling it to ramp up or slow down its own drug-metabolizing enzymes. They can also fine-tune our immune system, a discovery that has massive implications for one of the most exciting areas in medicine today: cancer therapy. The impact of pharmacomicrobiomics is being felt across a huge range of medical specialties. Let's look at oncology. The gut microbiome has emerged as a star player in determining who responds to cancer treatment. This is especially true for a revolutionary class of drugs called immune checkpoint inhibitors. These drugs don't kill cancer cells directly. Instead, they take the brakes off the body's own immune system, unleashing it to recognize and attack tumors. The results can be miraculous, but they only work for a subset of patients. Groundbreaking research has shown that the composition of a patient's gut microbiota is a major predictor of success. Patients with a diverse gut microbiome, rich in specific types of bacteria, often have a much stronger and more durable response to these immunotherapies. It seems our gut bacteria are crucial for priming the immune system to be ready for the fight. This has led to clinical trials where researchers are manipulating the microbiome through diet or even fecal transplants to try and turn non-responders into responders. The influence also extends to cardiology. Beyond the digoxin story, the metabolism of statins, the world's most prescribed cholesterol-lowering drugs, can also be influenced by gut bacteria. Even the gut-brain axis, the constant two-way communication between our digestive system and our brain, is heavily shaped by our microbiome. This has profound implications for neurology and psychiatry. Take L-DOPA, the primary treatment for Parkinson's disease. L-DOPA needs to get to the brain to be converted into dopamine, but we now know that certain gut bacteria can metabolize L-DOPA in the gut, essentially intercepting it before it has a chance to reach its target. This could explain why the drug's effectiveness can wane over time. Similarly, emerging research suggests that our response to common antidepressants like SSRIs may also be shaped by our microbial inhabitants, opening up new avenues for treating mental health conditions by focusing on the gut. Despite this incredible potential, the field is still young, and there are significant challenges to overcome before this science becomes routine clinical practice. The biggest hurdle is the sheer complexity and variability of the microbiome itself. Unlike your human genome, which is relatively static, your microbiome is a dynamic ecosystem. It changes based on what you eat, your lifestyle, your age, your stress levels, and the medications you take. This makes it a constantly moving target. Standardizing research methods is another key challenge, ensuring that findings from different labs around the world are comparable and reliable. But the future of pharmacomicrobiomics is exceptionally bright. The ultimate goal is to integrate microbiome analysis directly into patient care. Imagine this future scenario. Before prescribing a medication, your doctor takes a simple stool sample. This sample is analyzed to map out your unique microbiome profile. Using this data, an AI algorithm can predict how you will respond to different drugs. Your doctor can then select the medication that is most likely to be effective for your specific microbial makeup and tailor the dosage to maximize benefits while minimizing the risk of side effects. We could see the rise of microbiome-based diagnostic tests that act as biomarkers, predicting drug response before the first pill is even swallowed. We could also see more therapies based on microbiome modulation. This might involve personalized dietary plans, highly specific prebiotics that feed beneficial bacteria, or next-generation probiotics designed to enhance drug efficacy. In some cases, it might even involve fecal microbiota transplantation to completely reset a patient's gut ecosystem to make a treatment work. When we combine pharmacomicrobiomics, the study of our microbial genes, with pharmacogenomics, the study of our human genes, we get a truly holistic picture. By considering both our first and second genomes, we can achieve an unprecedented level of precision in medicine. In conclusion, the silent, teeming world within our gut is no longer a passive bystander. It is an active, influential partner in our health, a key determinant of how we respond to the medicines we rely on. The science of pharmacomicrobiomics is our new Rosetta Stone, allowing us to finally decipher the complex language of these drug-microbe interactions. As we unravel these secrets, we move closer to a future where medicine is no longer a matter of trial and error, but a precisely tailored strategy, designed not just for you, but for the unique microbial world you host. Thanks for tuning in to Fun Facts Online.